Good evening, everyone, and welcome to Science Cafe, November's Science Cafe. We are very fortunate to have Dr. Betul Kacar with us this evening. She is a NASA Space Fellow. That's a very prestigious recognition. And she also has a dual appointment at the University of Arizona in the Department of Molecular and Cellular Biology and the Department of Astronomy, because her field of research is astrobiology. She came to U of A. We're very fortunate to have her here a little less than a year ago, so she hasn't been in town that long. We're excited to have her here tonight, especially because she has a 5-month-old baby at home. So we're very grateful that she's taken the time to come and visit us this evening. She's a great speaker. I saw her speak once before. Really excited to have her. Please give a big round of applause for Dr. Betul Kacar.
Thank you. Thank you very much. I feel very fortunate to be here, and this is not how I've been spending my nights lately, so this is great. I am an astrobiologist, and some say that we are asking one of the biggest questions that people can ask, and I think it's true. We want to know, why are we here? How do we get here? How did we get here? Is there life elsewhere in the universe? And if we find life elsewhere in the universe, would it look like life that we have on our planet, or would it be completely different than what we have here on our planet? And I think what makes it really exciting is that these questions, even though they're really old, right? They're much older than us. We now have the tools that are available to us to study these questions in a scientific way. So we can develop hypotheses and we can test them, in my case, in the lab, and relate them to questions as big as, are we alone in the universe? So I'm going to take you guys through a brief time travel, so now walk with me. We are going to go all the way back. So we're looking at the timeline, and yes, humans are not even pointed here, because we've been here for a very, very brief time. I don't have a pointer, but we are really at the bottom, right after flowers and bees. And now we're going back. Our life is about 3.8 billion years ago. We think life on Earth originated around 3.8 to 4.1 billion years ago. And of course, we will talk about what I mean by life, whether that life looks like us today, or what did that look like. But now let's take a quick look and see what happened on our planet since the origin of life. So life originated. This is Earth. We think that some sort of heavy bombardment occurred. We can read these things when we look at the moon. The signs on the moon tell us that some sort of meteorite impact did take place around, I would say, 4.2, 4.3 billion years. And then we think that life originated around this time. Some sort of life, a very, very very simple single cell that originated around 4. When I have a music in the background, that makes it even funner. And then I will skip to about 2.5 billion years into past. We had rise of oxygen. So I will talk about this a little further, but we didn't even have oxygen on our planet for the first 2 billion years. So no oxygen, and then we have the great oxidation event, rise of oxygen. And then from 2.5 billion years, the rest is history, right? We have sponges and Cambrian explosion, the rise of all these animal species, another layer of oxygen, and then sharks and birds and mammals, and life is great. And as a part of the Women in Science series, I was asked to present you guys a woman figure that also impacted our field. And I picked a woman that made a significant impact in the way we understand the origin of eukaryotes. And that is Lynn Margulis. So Dr. Lynn Margulis, that we unfortunately lost in 2011, is the idea, she's the idea, the proposer behind the idea of endosymbiosis, that the host cell engulfs a smaller cell. So it's a day for a small cell to be eaten, to be a dinner of another cell. So she proposed this idea, and the story is that this is the paper that came out, I think it's in 1967 that she published this. I think this paper was rejected by about 15 different journals. And this is the 16th journal that accepted this paper, where she talks about maybe the way the eukaryotes evolved is a bacteria was a dinner to another bacteria. And of course, that's a very radical idea. And just to walk you through a little bit, she thinks that the chloroplasts originated as a free-living cyanobacterium and was engulfed by a protozoa, these are early animals, and reduced through time to metabolic slavery. So one animal gets together, one cell gets together with another cell and leads to a third cell that's completely different. And she hypothesized that the mitochondria even descended from an endosymbiotic bacterium capable of aerobic respiration. And I think what she really did was that she allowed us, I think a bit further and not just rely on paleontology when we think about the past, but also integrate biology as an idea when we think about where cells came from, how did cells happen to be. And of course, not so surprisingly, she was known to be a little, a crazy woman, but you have to be a little crazy woman to, to propose these ideas. And she changed the way we think of animals and origins of animals. So what do I do in my research group? I moved here in late January earlier this year. It's been an insane year, so it's funny to think that it's been only 10 months, I think, since I moved here. I came from Boston, from Harvard. Things are very different here. It was a great time to move here in January. We had a terrible snowstorm, and I think they have another one today, so yes. And look at this. So my lab is, we concentrate on reconstructing ancient biology. So I'm going to walk you through a little bit of how we do this. I tend to imagine that we are traveling in time in my lab every day. Yes, physically we are in Tucson, but our minds are billions of years in the past. And we combine a variety of different tools that we rely on evolution, molecules, we study genes, we study proteins, we focus on whole systems as well, such as bacteria, and then we try to tie this to the environment, to rock records. So yes, we combine geology, we combine microbiology, we combine systems biology and evolution, and ultimately we look at the stars and try to tie in what happened on our planet billions of years ago to potential presence of life elsewhere in the universe. So what is astrobiology? It's the scientific discipline dedicated to study the origin, evolution, distribution, and the future of life in the universe. So we are focusing on extraterrestrial life or its potential existence, but also trying to understand life on our planet. So when we think of life, I think we always forget life elsewhere in the universe. We forget that there's life in space, right? We are it. But how much do we actually understand life on our planet? Right? We're talking about life that's been spanning our planet for 3.8 billion years, at least. That's a lot of life. So how much do we know about it? And NASA is one of the foundations that invests a lot in understanding these questions. Thanks to your support, we can go and ask these really fundamental questions by combining a variety of different tools that come from a variety of different disciplines. So astrobiology is a very interdisciplinary field. It combines physics and cosmology, astronomy, astrophysics, biology, geology, chemistry. So we do get together, and most of the time, we don't understand each other, right? Because we all speak a different language. We all have our own disciplines, so it pushes us in a kind way to come up with our own norms of conversation, and we develop our own way of communicating, which is really necessary because at the end of the day, we all are interested in the same question. How did life originate? What happened on our planet to change the life the way it is, for it to be the way it is today? And does life exist elsewhere in the universe? And how can we define life? And how can we detect life? And how do we know that what we look at up in the stars is life, right? It's such a big question, yet it relies on life itself, which happens at the molecular level. So if you think about it, looking for life elsewhere in the universe is a molecular question. In one level, we have all this giant scale, and then we have to focus on a very, very small scale, a phenomenon that happens at the molecular level. So it is a clash of scales. So what is life? I guess Hollywood tells us it's this, right? If you go to Roswell, this is also it. I have a lot of stuff from that gift shop. I'm not ashamed of it. Would we be convinced if we find mushrooms on Mars that we found life? Or is it an elephant walking on Mars? Is that life? What is it? Well, I don't think it's this. One can believe. I want to believe. Yeah. But we think that life probably is something like this. microbes, right? So even though microbes dominated our history, we tend to ignore their existence, even though they made a significant impact. So when we think of the past or ancient times, maybe you're thinking of dinosaurs, right? Maybe you're thinking of a T-Rex, right? But for an astrobiologist, life really is microbe, because they really are the ones that occupied about 90% of our time, of life's history, was occupied by microbes. That's a lot of time. So they made significant impact and been present through history. And they even influence human behavior. You're looking at two of the historical figures, Napoleon and Lenin, who were thought to have a version of this strain in their system, which people think may have influenced their behavior. So microbes not only shape the environment and perhaps even led to oxygen that we breathe, but also changed human history in ways that, no comment there. All right, so we do live in a microbial world. Yes, please, this is great.
Saying that Napoleon and Lenin both shared intestinal parasite?
Yeah, there's a, this is I think more speculative, but it's been thought that they both were infected by this parasite, yeah. Yeah. But if there's one thing you remember from this talk, I hope that's not it. Okay. She talked about Napoleon and some, like, there's, okay, so we live in a microbial world, and there are about 1 trillion species of microbes on Earth. I think it's really, really amazing. I heard that there's more microbial species in the gut of a bumblebee than the human population on our planet. Yeah, that you can remember. So if you do not like bacteria, you live in the wrong planet. I'm sorry, move. This is it. This is what we got here. So we have all these crazy environments, right? You're looking at these purple lakes, and maybe you may recognize this from Montana, or desert, right? So all these environments that look so strange to us and may not be hospitable for us, they are hospitable for bacteria, right? Bacteria, microbes, find a way to survive under these harsh conditions. So when you think of like a thermal spring that you go, I like to soak, a lot of different bacteria, extremophiles, for example, that learned how to live in this hot condition that are not really nice for us to survive, right? In this lake, for example, it's very acidic, so if we were to get in there, we would disappear in a matter of seconds, but bacteria found a way to live in there. So we think that life started with a single cell, such as bacteria or bacteria-like organism, was the first being on our planet. That's one of the ideas, and that's the, I guess, the million-dollar question is that how did that first cell look like? How did that organism behave like, right? That is our ancestor, and we refer to it as LUCA, the last universal common ancestor. And we divide the history of time on our planet into 4 distinct groups. We are in the latest time, Phanerozoic, and Hadean and Archean are really very exciting for us astrobiologists, because this is where we see the rise of oxygen about 2.5 billion years, and life's origination is around here. And of course, no astrobiology talk is complete without quotes from Carl Sagan, but I like this one as well, from this book, wonderful book called Basin and Range. If you haven't read it, I highly recommend it. But here is what the author says, that if you free yourself from the conventional reaction to a quantity like million years, you free yourself a bit from the boundaries of human time. And in a way, you do not live at all, but in another way, you live forever. And I think thinking of time is a big component in the study of astrobiology, because we think of the billions of years of time scales. One also has to consider that the past bacteria survived under conditions alien to us. So when we think about alien life or our alien planets, I would like to challenge you into thinking that our own planet was an alien planet. Right? So let's look at the Hadean Earth. Hadean is a derived word from Greek, hellish planet. The first 500 million years is a crazy planet, right? It's really hot, lots of volcano, lava. We think there were a lot of meteorites crashing, lots of lightning. This is our planet birth. And life, we think, evolved in this condition. And the Archean Earth, then the next 2.5 billion years, is where we see the next 1.5 billion years, the Archean Earth, where rise of oxygen and the first bacteria and its descendants. Of course, the Precambrian era, the 500 million years, where we think that the species, the amount of species and the type of species have diversified and increased. And we refer to this as Cambrian explosion. It's not necessarily an explosion. It's not an overnight incident where there was no species and then there were a lot of them. It's a gradual increase in time. It's a long process. However, Earth's past can be viewed as a strange planet. It had different atmosphere. As I said, it had no oxygen. Nitrogen levels are also very different than what we have right now. Temperature, we think it was really hot, right? So different temperature, different atmosphere. And we have a lot of different planets, even in our solar system, that resemble our own planet's past. So Earth's past is a strange alien-like planet, but microbes found a way to live. So how did they survive under these conditions? Let's review this a bit further. We have the Hadean planet. And then here we are, the great oxidation event, rise of oxygen in the atmosphere from 0 to a little 1%. And Rodinia supercontinent is about 900 million years into the past. And the snowball Earth, where the whole surface of the planet was covered in ice. I think it's really crazy to think that our planet went through all these phases. And ice ages, these are more recent timelines. But as you can see, our planet resembled different stages across through its time alone. And these are just very hand-selected 5 different phases. So how do we look for life, and how do we understand what happened during that time? Well, as astrobiologists, we rely on biosignatures. And the way I always think about this is that if you sit on a seat for so long, and if you stand up, you see the shape on the seat, right? So that's your signature, right? It's the same thing, okay? For microbes, they sit on the rocks, and then they aren't there, and then they leave these shapes. Sometimes it's morphology, and sometimes it's chemistry. Sometimes it's chemicals that they leave behind. And by measuring them, we can get an idea about, first, their presence. And if you are a graduate student or a postdoc, looking through the desert or studying rocks and you find a fossil, congratulations, you just made it to the cover of Science. You found the rock, the fossil that says life maybe is as old as 4 billion years old. That's great. So it can be an object, a substance, or a pattern whose origin requires a biological agent. And this is a very important word, and I think our next generation will hear this a lot, because we are looking for biosignatures on different planets, and not only within our solar system, but outside of our solar system and exoplanets as well. As I said, there are different biosignatures, like morphology or shape, but I want you to think about the biosignatures that occur at the atmospheric level. So these are gases that a planet releases, if I may. And we can view the spectra that certain gases releases, speaking of Hadean Earth, and detect and understand what kind of gas does this planet have. So for example, let's look at our planet, right? If you are an alien civilization and you're looking for biosignatures and you are looking towards our solar system, you look at us. What do you see? Well, you see a blue dot today, right? It was not always blue. And if they have a similar technology, they would see all these different spectral properties for water and ozone and oxygen, carbon dioxide, whatever we have in our atmosphere is if you use the correct technology, is visible. So this is an atmospheric biosignature. Similarly, we can use different approaches to access the ancient biosignatures that are not necessarily at the gas level, but that could be at the microbial level. So these could be different chemicals that microbes release, or it could be different morphologies that they leave on the rock. So these are multiple papers. It could be at the molecular level. You're looking at these microbes that my colleagues collected from different environments, different soils. So you can see that they have color difference. And what we do is that we go to the extreme, strange environments today, and try to understand what happened in the past in these environments, or try to find an environment that mimics the past the best today on our own planet. However, the challenge is that all clades, meaning all of the current biology that are still alive today, represent less than 1% of all the species that ever existed on our planet's history. So we are trying to recover 99% of our history by relying on the 1%, and less than that is, that we have today. So talk about challenge. And if you're thinking of DNA, right, genes, it is even similar, well, at least the same proportion for the number of genes. And you will get that many of the DNA species, the DNA sequences that we have today, will be very different, that do not exist, that we just simply cannot sample. So then the question is, what is she talking about when she says she reconstructs biology? If they're all gone, right? That's a great question. That's what I do. We are using the current less than 1% available DNA sequences for the species available, and try to predict the ancient DNA sequences in my lab. So the question is, can we reconstruct the past? And the central thought is that microbes are going to be they are not only dominator of the past, but they will be what we find on another planet. So when we send a rover on Mars, that rover is looking for something like maybe a microbe, or a signal, or a biosignature that perhaps a microbe left behind, or a condition that may support the life of a single-cell organism. And this is a quote by Tim Lyons, who thinks really hard on these questions, that when we ultimately find a biosignature for life beyond our planet, it is almost certainly going to be microbial. And perhaps it will be very much like those described from the early Earth, our versatile test bed in one of science's greatest explorations. However, it's not just a microbe. I'm a molecular scientist. It is what the microbe entails. It's the machinery that's inside the microbe that makes it exciting, says the molecular biologist, right? We are interested in DNA and molecules, not just the organism itself, but getting inside the DNA. And this is not an easy task. Aside from the fact that many organisms are gone, we also are dealing with a complex problem. You're looking at a modern cellular pathway. So at any given time, all these reactions, I know it's not the highest resolution image out there, but this is the best I could do. All these pathways, all these arrows basically represent another reaction that happens at any given time in any biological cell. So this is a very complex problem, and really we are dealing with maybe something I can't even spot to, because it's very low resolution and there's a lot going on there. Yes. This is for modern cellular pathways for bacteria. Yeah. So, what did we do? First of all, I try to focus on the essential parts of a pathway. So we think that if life that we have right now shares components that are universal across all organisms, then they must be ancient. So let me put it this way. We all have a way, and by we I mean you, me, and a plant, a lizard, all the organisms have a way of processing information from the environment. And we do this through certain biological machineries, such as ribosome or translation machinery. So we think that the common molecular features of all life must be, first of all, ancient, two, essential, and probably universal, meaning that if all life on this planet shares the same exact molecule, life somewhere else probably will as well. So we make the simplest assumption that we possibly can.
What do you base that assumption?
What do you mean?
I mean, how can you assume that life somewhere else is like our life?
That's a very good question. So we refer to this as n=1 problem, that we have only one form of life, and that is planet Earth. So we base our searches mostly on what we can understand here. This is also in order not to waste any money, right? We try to find things that are similar to us and the things that we can understand. So we look for water, for example, because all life depends on water. So probably life elsewhere will need water. And if life we have right now shares common properties, probably an alien life will also have that too. These are very basic assumptions that we make. But yes, I agree with you. It's an oversimplification, but we work with what we can. But the question is, how do we travel back in time, right? So what do? I wish this is how I got into this. But it looks a bit different, okay? So we use something that's less fancy, equally complex, but it does not need a flux capacitor. I have to make this joke. All right, so how do we do this? This is a relatively new field called paleogenetics. So, and it relies on life's alphabet to make inferences about the past. So you're looking at an alphabet here. This is what we use, right, to communicate, to understand each other, basic alphabet. And this is what life uses. These are the bases that our DNA molecules, in this case RNA molecules, use. So, and if you were, I'm not sure, maybe we have a linguist here. Do we have any linguist? Anyone interested in? You are a linguist? Oh, that's awesome. Okay, so you will notice better than I do that if we try to reconstruct an ancient word, what do? We first look at the current cultures that exist today and try to find that word. In this case, you're now looking at snow. Okay, so we have the word snow here, English. It means different things. I'm not going to butcher this. I'm not German. Do we have a German here? Please be my guest. But it's Schnee, for example, in Bohemian and Serbian. It's Schnee. So we have different versions of the same word in different cultures, right? So the idea is then to infer the ancestral state of this word snow. And then what we do is, well, we predict a common ancestor for each of the node. And it turns out that the oldest ancestor, which belongs to Proto-Indo-Europeans, older than 3000 BC, it was something like Sni. Okay, this is the ancestor of the word snow, for the word snow. So we can predict the word, but also we can predict something else, right? We can predict that all these cultures that need to use the word snow lived in a snowy place, right? It was snowing. So they made up this word. So it's the same thing. So in my case, now we're changing these cultures with bacteria. It's the same thing. And instead of the word snow, now we have their own version. These are amino acids, right? So now we predict the actual ancestral amino acid the same way possible, right? And then come up with the ancestral version of this word. And the idea is that if all these organisms require this certain sequence that we usually know what they do at the protein level inside the cell, we can make predictions to understand the environment in which these organisms that needed this sequence, that this chain of words lives in. And yes, question? Right. So, in my lab, we focus on multiple fundamental components. This one is translation. I want to imagine this as a processing center, just like a computer. How all the computers, even the oldest, the most ancient ones that we don't have right now, have the similar processing center. Same thing with life. So we all have this thing. This is actually 3.8 billion years old. You're looking at the oldest molecule on our planet. This is it. And this is how DNA is transcribed and translated inside the cell. So DNA is made sense, if I may, using this molecule. And all life has it. I think it's insane that we all use the same thing. It looks the same, it smells the same, it behaves the same, we all have it. And also, we focus on fundamental mechanisms of how life uses carbon that's in the atmosphere, how do we generate biomass, how does life fix nitrogen. And these are interesting to us because these are the most essential and ancient phenomenons. Again, information evolved something around this time, right? So the way, it's almost the same, right? The way we understand each other, we speak the same language. So your brain, as I feed you words, translates this in your mind. The same thing for the cells. The machinery translates all this DNA information that it's fed to it. And if something doesn't make sense, usually there are problems, like cancer is one of them. Not the only reason, but definitely happens at that level. And usually cells has a way of fixing this, but it evolved really, really early on. And the great oxidation event was basically life's invention of making sense of the CO2 in the atmosphere and converting into oxygen, evolution of photosynthesis. So, what do we focus on? To summarize, we ask a lot of different questions. I did not talk about this today, but we also investigate the role of chance and necessity in evolution. Basically, the question is, if life were to evolve over again, would we even be here? Are we a result of random accident, or are we destined to be here? How much of life is shaped by deterministic forces? This is important for astrobiology because exactly we want to understand whether life elsewhere in the universe would be like ours. And that's a very, I think, also philosophical question. And what are the key factors of biology's past, and what is the likelihood of life's presence elsewhere in the universe? And this is a great time to study these things. This is a slide that a colleague of mine sent me from, she works at NASA. So you're looking at our solar system. All these blue dots are Earth-sized planets on our solar system. So what we're looking at, two things: size, Jupiter, Neptune, Earth, and Mercury. And you're also looking at the temperature, the surface temperature of these planets from red to blue. So blue is Earth. So what you're looking at now is that all the planets that Kepler could see, the telescope, that are present outside of our solar system. All right, so when you look at blue, you should be thinking, okay, well, we have examples of this on our own solar system. And the temperature, the blue temperature, is like our own. So they are almost Earth-like planets that are right at the outside of our doorstep. And just to give you an idea of what Kepler actually scans, so looking at the whole sky, Kepler looked at something like this. So that's how much of sky space that Kepler was able to detect, right? Very, very small. And look at all these planets. It's insane. And we didn't even think about the presence of planets that were outside of our solar system. Like even decades ago, we thought, come on, what's out there outside of it? Look at this. And this is not even all of it, right? So now we're building better telescopes to understand what's going on. In James Webb Telescope will be alive, hopefully, in a couple of years. And we're going to be able to scan even more. So I am very hopeful that, as I said, I want to believe that we will be able to find something promising after all these searches. And I think it's a great time to study these questions. And I think it doesn't get any bigger than this. So I feel very fortunate to be able to study this, even though, of course, the humble estimation is that everything we know, everything we see, and all the atoms in your body and in our galaxy, all the stars and dust and planets within and outside of our solar system and the universe, we really have no freaking idea. Okay, that's all I have. Thank you.